Okay, good morning all of you. In today's class, let's discuss about eukaryotic vectors. So far, whatever the vectors we discussed, majorly they were meant for prokaryotic host organisms. But there are many genetic engineering applications where you need to express these eukaryotic genes in different species of eukaryotic organism, because the proteins expressed will not be fully functional if you use the prokaryotic organisms as host. So that is why we are in need of the vectors which are capable of expressing the gene products in diverse species of eukaryotes. So now let's see what are the different eukaryotic vectors available, what are their properties, and how these eukaryotic vectors can be used in DNA cloning. Yeah. So before going to the properties of these eukaryotic vectors, when you talk about cloning in eukaryotes, as I mentioned, many eukaryotic genes and their control sequences, they had been isolated and analyzed using the cloning techniques based on E. coli as host organism. That work had been done, but still there are many applications of genetic engineering, uh, like for example, from the large scale production of eukaryotic proteins to the engineering of plants, animals, and gene therapy. Such applications of genetic engineering that requires the vectors for the expression of genes in different eukaryotic species. As I told you, if you express these genes in prokaryotic organisms, the proteins produced will not be fully functionally active. So we need the vectors uh, by which we would be able to express the proteins in different eukaryotic organisms. That's where there was a need for developing the vectors for eukaryotes. Before going to the different types of vectors and properties of these eukaryotic vectors, I would like to discuss briefly about the transfer methods of gene transfer in eukaryotic cells. What are the different methods of gene transfer that can be applied to eukaryotic cells? The take-up of DNA into eukaryotic cells is called transfection. Whereas in case of prokaryotes like bacteria, E. coli, we refer to it as transformation. But the take-up of DNA into eukaryotic cells, the method which is called transfection, it is more problematic than bacterial transformation. The process is complicated than bacterial transformation. And moreover, the efficiency of this transfection is also much lower than the efficiency of transformation. So if you take yeast and plant cells, what you need to do is, because these plant cells and yeast cells, they consist of cell wall. So first you need to digest the cell wall with the degradative enzymes. When you digest the cell walls with degradative enzymes, the protoplasts are exposed. Then these exposed protoplasts would be able to fairly take up the DNA. So when your eukaryotic cell is, if it is a yeast or plant cell, since they have cell wall, first you need to digest these cell walls by treating these cells with the degradative enzymes so that their protoplasts are exposed, and this exposed protoplast will be able to take up the DNA readily. And the cell walls can be later resynthesized. Once you remove the degrading enzymes, then the cells would be able to resynthesize the cell walls. But if your target cells are animal cells which are maintained in culture, in that case, we all know that animal cells do not contain cell wall, isn't it? So in that case, they will take up the DNA at low efficiency if the DNA is precipitated on the cell surface along with calcium phosphate. This method is called calcium phosphate precipitation method, which is used for animal cells. If you want to transfect the animal cells, you can precipitate your DNA of your interest along with calcium phosphate. Then if the cells would be able to take up the DNA with very low efficiency, and as I told you, this method is called calcium phosphate precipitation method. And I mentioned that the efficiency of this process is very low. So you can increase the efficiency of this process by treating the cells with high voltage current. When you treat the cells with high voltage, what happens? That opens up the pores in the cell membrane. When the cell membrane pores are opened, then the take-up of DNA by these cells will be done with better efficiency. This process by which the cells are treated with high voltage current, by which the cell pores in the cell membrane are opened, by that process the efficiency of transfection will be increased. This whole mechanism is called electroporation. So we have discussed so far, in case of the yeast cells containing cell wall, we treat those cells with the degradative enzyme so the exposed protoplast will be able to take up the DNA fairly. If they are animal cells, as you know, animal cells do not contain cell wall. In that case, the methods that are used could be calcium phosphate precipitation method. Of course, with this calcium phosphate precipitation method, the efficiency is low. To improve the efficiency, we can treat the cells with high voltage current so that the cell membrane pores will be opened up. This process is called electroporation. Then there are also few direct physical methods available for the gene transfer in case of eukaryotic cells. What are they? You can micro inject the DNA. The target DNA can be micro injected using very fine glass pipette. It could be directly into the cytoplasm or even to the nucleus of animal or plant cells which are maintained in culture. So the physical method which can be used for gene transfer in eukaryotic cells could be micro injection, where a very fine glass pipette is used to directly inject the target DNA either into the cytoplasm or to the nucleus of the individual cells which are maintained in culture. One more physical method that's available is gene gun. In this gene gun method, the DNA could be introduced by firing metallic microprojectiles. These metallic microprojectiles are the metal particles which are coated with DNA, and these can be shot at the target cells. This method is known as gene gun. These methods are very important methods of gene transfer to the eukaryotic cells, or methods of transfection. Whatever I listed so far with yeast cells, plant cells, and calcium phosphate precipitation, electroporation, micro injection, gene gun, these are all the various methods of transfection. This topic is given as one of the seminar topics, so I'm not going to the more details. Just the brief introduction I want to give before going to the shuttle vectors. Later it will be covered a little bit more in detail when the seminar topic comes. Now let me move on to shuttle vectors. What are these shuttle vectors? Most of the vectors whichever had been developed for eukaryotic cells, they are constructed as shuttle vectors. These shuttle vectors, they incorporate the sequences that are required for prokaryotic organism like your E. coli, and also the sequences required for desired host cell. Here the desired host cell could be eukaryotic cell. So what I am saying, these shuttle vectors include two types of sequences. One type are the sequences which are required for replication and selection in E. coli. Since it has the sequences for replication and selection in E. coli, these vectors enable the construction of recombinant DNA molecule, and its integrity can be checked using the E. coli methods. As you all know, the E. coli methods like the plasmid methods are well developed and characterized. So it becomes easy. The initial steps of this cloning process would become easy when you use these shuttle vectors, because the construction of the recombinant molecule and checking its integrity can be carried out in the prokaryotes itself, which is comparatively easier method. Once that process is done, where you require this eukaryotic cell expression is only during the expression step. That means to express the proteins which are functionally active for eukaryotes. So before transferring to the eukaryotic cell, this initial procedures can be carried out using the E. coli methods. Then it can be transferred to the appropriate eukaryotic cells, because these vectors also include the sequences that are required for the desired host cell. So after this initial process of cloning, it can be transferred to the appropriate eukaryotic cell uh, in which the vector will support the expression of proteins. So one such shuttle vector we can take as example and discuss is yeast episomal plasmid. Yeast episomal plasmid. If you remember, even the ACT vector, yeast artificial chromosome, is also a shuttle vector. There, when we discussed about its properties, it has the plasmid sequences and also the sequences that support the cloning in eukaryotic cells like your yeast cell. So those also come under the shuttle vectors. This is another example of shuttle vector, yeast episomal plasmid. So this is the vector for cloning and expression of genes in Saccharomyces cerevisiae, which is designed based on the natural 2-micron plasmid. This yeast episomal plasmid vector had been designed based on the naturally occurring 2-micron plasmid. This 2-micron plasmid is named for its length, because the plasmid length is 2 micron. That's why it's called 2-micron plasmid. And the size of it is 6 kb. This 2-micron plasmid, it has an origin of replication. What are all the sequences present on this 2-micron plasmid? It has an origin of replication, and there are also 2 genes present which are involved in replication. In addition, this plasmid also encodes a site-specific recombination protein FLP. This FLP protein is homologous to the Phage integrase protein, that is INT. So these are all the sequences that are present on this 2-micron plasmid. So the vectors based on this 2-micron plasmid are called yeast episomal plasmids. And you all must be aware of it. Episomes are the plasmids. They have the property of getting integrated with the host chromosomes, isn't it? So something similar you would see in case of this yeast episomal plasmid vectors. So look at here, look at this diagrammatic representation of one of the example of YEP vector. So here the sequences present are, as I told you, it is developed based on the naturally occurring 2-micron plasmid. So there is 2-micron origin of replication, and there are also shuttle sequences. The shuttle sequences include E. coli origin of replication and the selectable marker for E. coli, that is ampicillin resistance marker. And this episomal plasmid vector also includes a selection marker for yeast. So here in this example, the selection marker is the LEU2. This is the gene that enables the biosynthesis of amino acid leucine. Here you can incorporate your gene of interest, that is gene X. So here this 2-micron plasmid vector or yeast episomal plasmid that can replicate as plasmid from 2-micron origin, or otherwise it can integrate into the yeast chromosome. So here what happens in the yeast genome also, when you take the auxotropic mutant of the yeast strain, there is mutant LEUT gene. So with the homologous recombination, uh, this vector can get integrated to the yeast chromosome. When it gets integrated to the yeast chromosome, your gene of interest is constructed on this vector, so it will also get integrated to the yeast chromosome. So that's how the cloning is carried out using this 2-micron plasmid vector, which is yeast episomal vector. So to brief it once again, this yeast episomal plasmid vectors are developed based on the naturally occurring 2-micron plasmid. It has the shuttle sequences like E. coli shuttle sequences and also 2-micron origin selection marker. It can replicate as plasmid because there is presence of 2-micron origin, or it can get integrated with the host chromosome, that means yeast chromosome, by homologous recombination. When it gets integrated with the host chromosome, the gene of interest also gets integrated. So it can be available there for the expression of the gene. So normally they contain 2 micron replication origin and also E. coli shuttle sequences, and a yeast gene which can act as selectable marker. In the example shown, the selectable marker is LEU2, which is involved in leucine biosynthesis. As I mentioned, though they can normally replicate as plasmids, X may integrate into yeast chromosome by homologous recombination. So that's about shuttle vectors. With one example we have discussed, that is yeast episomal plasmid. Next, eukaryotic vector exclusively for plant cells is Agrobacterium tumefaciens Ti plasmid. As you all know, the plant cells, they don't contain natural plasmids which can be utilized as cloning vectors. But there is one bacterium called Agrobacterium tumefaciens. This bacteria infects dicotyledonous plants, and this plasmid, it contains about, this bacteria contains the 200 kb length Ti plasmid. This Ti plasmid is tumor inducing plasmid. So what happens when this bacteria infects the plants? um, the bacteria contains the Ta plasmid. The part of the Ta plasmid called tDNA. Please follow. The tDNA is the part of the Ta plasmid. It has the property to get integrated with the plant chromosomal DNA. When the Ta plasmid tDNA gets integrated with chromosomal DNA, what happens? It causes a disease called crown gall disease by producing or by developing the tumors. I hope you are able to follow me. There is a bacterium, Agrobacterium tumefaciens, that contains the Ta plasmid. This is tumor inducing plasmid. A portion of this Ta plasmid called tDNA that has the property to get integrated with the plant chromosomal DNA. When it gets integrated with plant chromosomal DNA, it induces tumor, a disease condition known as crown gall disease. So you look at the action of this Agrobacterium tumefaciens Ti plasmid. As I told you, it is present in Agrobacterium tumefaciens. This is the Ti plasmid. Excuse me, I think one of you have unmuted your mic. It's causing disturbance. Please mute your mic. Yeah. So this diagram shows the action of the Agrobacterium tumefaciens Ti plasmid. As I told you, a portion of this Ti plasmid, tDNA, that gets integrated with the plant chromosome. When it gets integrated with the plant chromosome, the genes present in this integrated tDNA, they induce crown gall disease. So the property of this Ta plasmid can be made use for recombinant DNA technology. How? The recombinant Ti plasmids can be constructed with the target gene by inserting this target gene into the tDNA region. When your target gene is inserted into the tDNA region, that can integrate the gene into the plant DNA, thereby it can be expressed in the plant. So the idea is, since it has the natural property of integrating with the plant DNA, you can insert your gene of interest in the tDNA portion. So when you allow it to infect the plants, along with the tDNA, the gene of interest can get integrated with the plant genome, and there it can be expressed. But in practice, several modifications are done to this simple scheme, because the size of the Ta plasmid is quite large. Because of the large size, it becomes difficult to manipulate. So later they discovered that if the tDNA and the remaining portion of the Ta plasmid, if they are present as the separate molecules within the same bacterial cell, still the integration can take place. I hope you are able to follow. because of larger size of Ta plasmid, what they uh, analyzed and understood was that instead of having the Ta plasmid as one whole molecule, they separated the tDNA portion as one separate molecule and the remainder of the Ta plasmid as another molecule. And it was found that if these two portions of the Ta plasmid are present separately on two molecules within the same bacterial cell, still the integration can take place. Based on this discovery, what they had done is the recombinant tDNA was constructed. The tDNA of the Ta plasmid is separated, right? On that molecule, the gene of interest is incorporated using the standard E. coli plasmid techniques, and then it is transformed into the Agrobacterium Ti bacteria containing the remainder Ta plasmid without the tDNA. So that bacterial cell will contain the tDNA into which your gene of interest has been incorporated and also the remaining portion of the Ta plasmid. And further improvement is also done to this plasmid by deleting the genes of crown gall formation from the tDNA. I was mentioning in the uh, previous slide, the genes which are present in the tDNA portion of this Ta plasmid are responsible for causing this crown gall disease. To simplify, because we don't want that disease to happen, at the same time we want to use this Ta plasmid for producing the recombinant plants. So the genes which are causing this crown gall disease also had been deleted from the tDNA portion. So the so-called disarmed tDNA shuttle vectors can integrate the cloned genes into the plant genome, host plant genome. So if the host cells are grown in the culture, those transformed clones can be developed as complete recombinant plants by treating with the appropriate hormones. A schematic representation is shown here how the recombinant plants can be developed by using these Ta plasmids. So these are the plant cells that are maintained in culture. If you infect the Agrobacterium Ti bacteria containing your disarmed tDNA vector with gene X, this disarmed tDNA vector carries your gene of interest, and that same bacteria will also have another molecule of DNA containing the remaining portion of your tDNA. Then only the integration can take place. So when you infect the cells in culture with these bacteria, the cells are transformed. These transformed clones can be regenerated into a complete plant by treating with appropriate hormones. So by following this procedure, you would be able to produce genetically engineered plant with the gene of with the integrated gene of interest, that is gene X. That's how this Ta plasmid vector can be used to carry the genes of interest into the host plant. By that process, you would be able to produce genetically engineered plants. Of course, while discussing the last unit, applications of recombinant DNA technology, one separate topic is there, genetically engineered plants. There again, we'll be discussing more in detail how to produce these genetically engineered plants. There again you will come across this Ta plasmid, because there the vector that will be used for transferring the gene would be the Ta plasmid. So this is all I wanted to discuss in today's class. In few of the eukaryotic vectors, the requirement for eukaryotic vectors we discussed. Then before going to the few types of eukaryotic vectors, I also briefed about the methods of transfection into eukaryotic cells. I discussed about plant cells, yeast cells. Then in case of animal cells, we follow the calcium phosphate precipitation method, then electroporation, uh, then direct methods like micro injection, gene gun. These are all the methods of gene transfer into eukaryotic cells. Then I discussed about shuttle vectors. They contain the shuttle sequences for the two different organisms, or prokaryotes and eukaryotes. One such example I discussed was yeast episomal plasmid. which is developed on naturally occurring 2-micron plasmid. Then I also discussed about another eukaryotic vector, that is Ti plasmid, which can be used for plant cells. I hope this class is clear for you. In the exams, shuttle vectors are used for 5 marks. It has been asked for 3 marks also. And the Ti plasmid is very, very important for 5 marks, and the same Ti plasmid will be useful when you explain the essay question of genetically engineered plants for 15 marks. And another possible question from this topic would be gene transfection methods. That also is important question to be asked in the exam. So that is all I wanted to discuss in today's class. These are the references. Thank you very much for following the